From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 13, recorded on November 3rd, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code MICROBE. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels Elde.
Great to be here, coming to you from Elde Lab Studios.
You decided to go back home, huh?
Yeah, it's good to come home from time to time after sort of trotting around. Last time I was in your neighborhood at World Headquarters.
That's right. Our last episode with Josh Drew was here. And in the meanwhile, between then and now, you've been elsewhere. I know that.
Yeah, a few trips, including one out to Chicago last week, where I had a great chance to catch up with today's guest, who we'll introduce in a second here. And then back here, though, at least to catch my breath for a little while before going back to the road.
So the original plan was to do this episode from Chicago. You were going to be there, actually. And
yeah, exactly.
That fell through the cracks, but here we are, and we'll get it.
Get it to work. And sort of kind of inspired by hanging out with Josh Drew last time, and thinking a little bit more about ecology and field work. I was going to try to take sort of a page from his book and do a field trip to the Field Museum to record our this episode that we're doing now. But of course, my schedule was overbooked, and I ended up that didn't happen. But here we are.
Yeah, I don't think. Will you learn? Will you ever learn? Or no? You probably won't, right?
No, I'm hopeless with scheduling and double booking, overcommitting all these things.
Okay. Well, our guest today is, as you heard, from the Field Museum of Natural History in Chicago. She's the MacArthur Associate Curator of Insects, part of the Integrative Research Center in the Department of Science and Education. Corrie Moreau, welcome to TWiEVO.
Great, thanks. I'm really excited to be here.
Yeah, we're very happy you found the time to do this, because we got really interested in your cool work there, and we're going to talk about it and talk a little bit about you too. So thank you so much. It's cool. We were just having a pre-show conversation about more and more involvement of museums and the stuff that Nels and I do. Molecular biology. It used to be just dusty collections, and now it's totally different. People like you are working there, and others as well, and really making incredible advances. So I want to give our listeners a little hint about that today.
Sure. And I mean, I'm pretty excited too that I think that people are starting to begin to appreciate the knowledge that's locked behind the doors of natural history collections the world over, and sort of the big picture questions we can begin to tackle.
Nels, who is it we talked to that, I guess it was Joss, he said in one museum that they wanted them to do their experiments behind glass and let people watch them? Do you remember that?
Yeah, that's right. And there are a few examples of that, right? Is there one locally by you, Corrie?
There's actually one in the Field Museum, and in fact, Josh Drew was a postdoc that worked in our DNA lab. So our DNA lab is behind glass so that people can see us working in real time.
So that's what he was talking about, right? Yeah.
That's right. I think one of the biggest outcomes of putting things like research laboratories behind glass in public museums and potentially other places where people travel has really been that it breaks down the barrier about what people think stereotypes of scientists are. I mean, I was really excited that I'm like, I'll get this great opportunity to share all of the cutting edge research we do. And we definitely get to do that. But I think one of the things that's sort of the most empowering for guests that come is that we just look like them. And I think that's really eye-opening for them that, we're scientists and, anyone can be a scientist.
So one of the things we do on these podcasts, I do 5 different podcasts, and one of the things people like is that they get to hear scientists talking. And we don't just talk about science, we talk about the weather, what we do, our problems, this and that. And they love it because they say, wow, we can see that scientists are people like us.
That's right.
That's right.
And you take it one step further. You let them watch you, which I think is really good too. So all this is really good, and it's taking advantage of being able to communicate.
That's right. And I would say, too, we were just talking a bit about this, that not only for the guests who come to see, some of the scientists in action, but for scientists to come to these museums from all different disciplines and fields, and to feel like this is just these beautiful spaces celebrating, broadly speaking, what we do, our profession. And so for me, going to a place like the Field Museum or Natural History Museum, we have a great new one here in Utah. It's just like an oasis to sort of refresh and rejuvenate, and to get some inspiration for the things that we might do in the laboratory by drawing from nature all around us and sort of how we broadly sort of categorize or catalog these things.
Yeah. So, Corrie, let's start at the beginning with you and tell us a little bit about where you're from and all your training you went through to get to where you are today.
Sure. So I'm actually from New Orleans, Louisiana. So I grew up in a place that's hot and sticky and full of bugs, which I think is what sort of, led me down the path of being a, tropical field biologist who collects bugs. But I actually knew at the time that growing up that I wanted to sort of spread my wings and, fly the proverbial coop, so to speak. And I actually went and did my undergraduate degree at San Francisco State University, which actually was an amazing experience for me on many, many levels. I mean, being in San Francisco was great, but I had a really great group of faculty advisors and fellow students who I'm still very close with from San Francisco State. I then stayed there and did my master's degree, but did it in collaboration with the California Academy of the Sciences, which is the Natural History Museum in San Francisco. And that was sort of my first exposure to really being able to use the scientific collections in museums. I mean, we had a small entomology collection or museum in San Francisco State, but it was modest in size and it was very much local organisms where, being able to take advantage of these world-class collections that are essentially from every part of and corner of the world was really in some ways liberating for me to sort of think that there are no boundaries, both in space but also in time. Like we can start to ask questions that go deeper back in time than just what we can collect today. And then that for me really compelled me to think about, well, I'm interested in using molecular and genomic methods, but I want to couple that with, biological collections. And so where's the best ant collection in the world? It's at Harvard University. So I actually went there to do my PhD, which was a really fun time for me, both because of just sort of the intellectual freedom that I had, but I got to be advised by two amazing mentors. One was Nomi Pierce, who's done a lot of work on butterfly ant interactions. And so she had a really unique perspective on thinking about ant evolution, mostly from the butterfly perspective. But then I was also co-supervised by E.O. Wilson, and so he clearly is, the grandfather of ant biology, and so really having those two as champions behind me really pushed me to think big.
Yeah. You have some pictures. I guess he visited the museum. You have some pictures on your website of him, right?
That's right. Yes,
that's pretty cool.
He's been really great mentor to me throughout my entire career. And then I went and did a Miller postdoctoral fellowship at UC Berkeley before starting my position here at the Field Museum 8 years ago.
So are you, do you know only about ants or do you know all bugs?
I have an undergraduate degree in entomology, so I know about all bugs, but I've spent a large portion of my career trying to understand everything about ant evolution and all of the things that interact with them, whether it's the environment, whether it's their gut bacteria, whether it's the things that they're eating. I've spent a bit of time thinking about that.
So if I showed you a picture of something, would you be able to identify it?
I hope so.
Okay. So
Vincent, are you worried about a recent bite or something related?
I have always wanted to do a bug podcast, and I've always been looking for someone to do it with. And so I'm just probing here, because
there's an ulterior motive.
Well, I got to tell you, you have a great voice. You speak really well. Your science is really good. So if you know bugs.
Excellent. So I didn't realize this was an interview at the same time being an interview.
It wasn't. I think a bug podcast would really be cool. People would love it because I think everyone's fascinated by bugs, and it's just a matter of finding. I talked Nels into doing a an evolution podcast. So
maybe offline I should ask Nels what he thinks about this.
Yeah, you can. So if someone asks you, what do you call yourself? What word would you use? Would you call yourself an entomologist or something else?
Gosh, that's a tough question because I think it depends on the audience. So I most frequently say I'm an evolutionary biologist, but I also say that I'm an entomologist, and I say that I'm a myrmecologist, which is somebody who studies ants. I also say that I'm a systemicist, and that I mean, there's all kinds of things. A phylogeneticist. I guess somebody who studies molecular evolution. I mean, I kind of do a lot.
Okay.
Yeah, and I think you kind of put your finger, Corrie, on maybe a lot of scientists coming up these days, that it is really hard to sort of even identify yourself in one setting or another, because so many people are kind of reaching across disciplinary boundaries these days. And so one of the things that's really fun about our profession, right, is that we can wear these different hats or explore in different territories. And
I absolutely love it. I mean, I feel like, at least for me, during graduate school, you spend quite a bit of time becoming an expert in one thing. And so you start out, very excited and interested in lots of questions. But just by the nature of doing your dissertation, you spend, several years focusing in on one question to sort of really be able to tackle it well. And then when you pop out the other side, suddenly you have this opportunity to expand again. And for me, that's been, I think, the most exciting time, and it's from borrowing from different fields. So starting to think about, the microbiology that's associated with, the hosts that I'm looking at, or thinking about how we can use things like stable isotopes to infer sort of the trophic position, which is a proxy for diet, to understand how that's shaping the bacterial communities. And then what role does symbiosis have on genome evolution? And what can we say about interactions of these organisms in nature? So thinking about ecology. And I think for me, that's where I get really excited, is instead of spending time doing only one thing, as a community, we're embracing that we really truly have to be integrative now.
It's funny that this comes up because yesterday I spoke with an NPR reporter who wanted some comments on a virus story, right? So she says, just state your name and your title. So I said, I'm VR. I'm in the Department of Microbiology and Immunology here. And she said, "Oh, you mean you're not a virologist?"
Yeah.
I said, Let me restate that. I'm a virologist at Columbia University. And she said, it's so hard to get people to say what they actually do. And that's because you can't, right?
That's right.
Exactly.
That's right.
It's so funny. I thought, microbiology is, a super family of viruses, but it's the same question. So I think we got up to your postdoc. I don't remember.
And then you said, Corrie, that 8 years ago you landed at the Field Museum. And what was it that brought you to Chicago?
So I think for me, when I was in my last year of graduate school, I was, like most graduate students, thinking about, postdoc opportunities. And I actually had no intention of applying for jobs because I didn't think I was quite, prepared enough yet. But this position opened up and I thought, wow, a world-class institution that has a major collection with great ties to the University of Chicago. I mean, I'll throw my hat in the ring, but as a graduate student, I won't likely be considered. And so still applied for lots of postdocs. And so actually the first time I ever came to Chicago was to interview for the job here. And I mean, if you've ever been here, it's such a grand building. It's just the building itself is so immense that it's almost overwhelming. And then, we have, 35 million specimens and artifacts behind the scenes to do science on. And so I think for me, that's what really sort of drew me here. And then, eventually I got offered the job and they were really generous enough to let me defer to go do a postdoc at Berkeley first so I could feel more confident and prepared to be able to run my own lab.
Ah, okay. That's really nice. You could do
that.
So what was it? They don't have a lot of ants, isn't that? That's not what attracted you, right? Just the overall collections and opportunities there.
Right. So, they do. We actually have, probably 1.5 million ants in our collection. I'm still adding to those all the time, but you're right. We don't have the largest collection by any means, but I think it's the culture of appreciating field biology through organismal biology, through molecular biology, and even the fact that I, had these crazy ideas about doing, microbiome research on, these specimens and collections, which hadn't started here yet. And the fact that they were like, that sounds really great. Like, let's do it. And so I think it was just that attitude towards, we might think about the questions we tackle from an organismal perspective, but we welcome having the opportunity to sort of come at it from many angles.
So these 1.5 million ants that you mentioned, these are all pinned or stored. They're dead, right?
They're all dead. That's right.
And so, and you add to them, so you go out and collect more. Is that right?
Yeah, that's actually, I think, one of my favorite parts of my job is that I've been able to maintain an aspect of field biology to my research program. I still get to go and actually go to the field and collect bugs, which I love.
Where do you go?
So this year I went to French Guiana in South America. I went to India. I've been to the Florida Keys. And then is that all for this year? Maybe one more place that I'm forgetting. Oh, I went to Mozambique.
Yep. Yeah. So Vincent, we were talking a little earlier about how sometimes we get caught up in the whirlwind of travel. I think it's fair to say Corrie has us both beat hands down with the locations that she traverses from year to year.
Yeah. Well,
and actually, so if I'm remembering right, Corrie, I think we met first in person in Potsdam, Germany.
That's absolutely right.
Yeah. So there was a meeting from the Kavli Foundation coordinating with the national academies, and then these bicameral meetings, in this case with the Alexander von Humboldt Society in Germany. And so I ended up organizing a session on the microbial control of host behavior. And Corrie came to talk about some of her work on ants and some of the fungus that can zombify these things, along with her colleague David Hughes. It was really a fun meeting.
It was fun, but I have to say maybe I'm biased because it was all about like microbes in organisms, which I absolutely find fascinating.
It's funny. When I need a virus, I just send for it, and when you need an ant, you go get it.
Yeah.
Maybe I need to revise my...
That's right. In fact, I have a new PhD student who we're just starting to think about the influence of viruses across ants and thinking how it might shape their evolutionary history. So maybe we'll be knocking on your door at some point. You can join us on a trip to some far-flung place.
Oh, I would love to do that. I'll take my camera and stuff and make a movie out of it. So how do you decide... I want to know about the nuts and bolts here. I want to know how you decide where to go, and once you're there, how do you actually find and collect and bring them back?
Yeah, so it depends on the specific research question we're tackling. So we currently have a grant from the National Science Foundation to study the bacterial community associated with a group of herbivorous ants, and they're called turtle ants, and they're from the genus Cephalotes. And these ants are distributed there's 3 species in the southern U.S., but most of their diversity is from Central America through South America. And so for that grant, we actually want to be able to understand something about what structures gut communities both within species and also among species on an evolutionary scale. So in that case, we want to actually travel to places where we have more than one species coexisting, because the question is, how are these microbes sort of shared among partners, and how often do they switch? So in some cases, these species, you can have multiple species living in a single tree. They're all arboreal. And so you can imagine that might be a mechanism for microbe swapping among hosts. And so really what we want to do is we want to sort of survey the species across its known distributional range, see how many of those microbes are sort of constrained to hosts' evolutionary history or phylogeny, and then how often do we get those exchanges happening, and is it correlated with proximity? So if you have 2 species living in the same tree, is that a higher chance that you're going to get, horizontal transfer of microbes or not?
Mm-hmm.
So in that case, we do a lot of work in Central and South America, and so that's where I've been doing a lot of research. But I have students working in India and in China, and so in those cases, I get to go and join them and be their field assistants, which is really fun.
So if you want to sample ants in a tree, you have to go up the tree, right?
In some cases you do. So that's actually one of the most fun parts, I think, for me lately, is being able to take advantage of these systems that let you access the canopy. So I've done canopy access systems in many different forms. There are lots of ways to get access to the canopy, but I've done a canopy crane in Queensland, Australia. I've done some of these canopy walkways. I've done canopy towers in Ecuador and Peru. But most recently, the most interesting one that I've ever done is this thing called COPUS, which is this canopy access system in French Guiana, which essentially is almost like a zip lining chair that you just sit in, and there's a crane down on the ground that controls all the mechanics, and you have a little remote control that you just drive yourself around in. Wow. And since all the mechanics are way down on the ground, it's absolutely quiet up there. And so you're just driving yourself through the canopy and interacting with all these organisms that are not, scared away by all the noise. It's amazing. So
how do you actually collect the ants?
Yeah. So most of the ants that I study that are arboreal actually live inside hollow twigs. So you spend a lot of time snapping twigs. Sometimes you'll see them running around on a tree, so you know they're there somewhere. And so I spend a lot of time snapping twigs and throwing bits of twig that have live ants in them into plastic bags or tubes, or so that we can process them when we get back down to the ground.
And so how many would you collect from a given tree? A few hundred or dozens?
Yeah, it depends on the project. So in some cases, when we want to do things like diet manipulation experiments, we actually need to collect hundreds of them so that we can essentially take that sample and split it into multiple iterations so that we have controls, and then we have different diet experiments we're feeding to see what influence diet has on bacterial gut communities.
So in most cases, you're going to kill the ants at some point. You're not going to bring them back alive, right?
We have brought them back alive depending on the project. So in that case, sometimes we do. Most of the time they're actually dead. And so we pickle them in usually 95% ethanol, sometimes RNA later, sometimes in liquid nitrogen, depending on the project. Yeah.
And you how do you identify them? You take pictures and look in books, or do you sequence their genome, and how would you do that?
So we typically try to identify them from their sort of anatomical or morphological characters first, at least at the highest level. In the field, you can more or less tell all the ant genera. In many cases, there's about 335 ant genera. In many cases, you can tell some of the species apart, but most of them you have to look at them under a microscope. And even then, as you can imagine, that the amount of variation is not high, and so discerning between different species is difficult. And we can use, DNA barcoding or genomic sequencing if needed.
How many different ant species do we know of globally?
There's about 15,000 species that have names by scientists. The number's at least double, if not triple. So there's more ant species than all the birds and mammals added together.
But not more than beetles, right?
Not more than beetles. Beetles have everything topped.
That's right.
Well, I don't know. I'd say viruses have everything topped.
Well, viruses might, but the being able to formally name them is a little different.
Yeah. Oh, this is cool. This is so cool.
No, I think a podcast on insects could really get some traction.
Oh, just imagine. So we could talk about ants probably for a year.
That's true.
If we did one a month, and then we could do stick bugs. We could do oh man.
Well, and even look, and then we even look at This Week in Evolution. How many of our episodes have ended up in insect space, whether it's some of the old domesticated fruit flies or some of the more, the peppered moths, the butterflies.
Yeah. Oh,
it's a lot of interesting biology unfolding.
There is a lot of interesting biology. And, I think that's something that people often don't appreciate. I mean, even within fruit flies, there is remarkable diversity, both in the morphology or the anatomy, but in their behavior it's striking.
Yeah, agreed.
Go ahead. Sorry.
Well, I was just going to say, one of the themes that's come up in sort of repeatedly in our episodes and from our listeners has been this idea of behavior and how do you study the evolution of it. And I think what we've kind of come to, at least as one idea, is that it really is the insects where you might have the possibility of having systems that are experimentally tractable enough to make real progress. And then for me, and for me, we've talked about this some too, it's the microbes manipulating the behavior of these insects, where now you have the microbial populations, whether it's viruses or bacteria, as almost these natural experimentalists who've done the work for us. If we can just figure out the rules by how the microbes impinge on the insects, we can really go a far way potentially in figuring out some of the molecular basis of behaviors, the evolution of behavior.
That's right. And I think that's one of the keys, is that there's so much diversity in the insects, both in the number of species, but also in the ways that they're either manipulated or that their behaviors are convergent or divergent. And so you have the sort of power to ask the question from the standpoint of multiple independent evolutions of something to say, what's driving it? Is a shared mechanism? Is it a co-opted, system or framework, or is it something novel every time?
Yeah, no, great point, Corrie. And that, nature's done the experiment so many times that's there's a huge advantage to that sort of repetition unfolding.
That's right.
Corrie, do you have a favorite ant?
Oh, of course I do. My very favorite ant is the Florida turtle ant. It's called Cephalotes varians, and you should absolutely look this ant up, because the reason that they're one of my favorites is they're beautiful to look at, but on top of it all, they have some really interesting biology and behavior and gut microbes. So it's kind of like it's got a little of everything that makes me happy. But one of the things that I think is really striking about turtle ants is they come in two forms. The worker caste does, where you have the small miners that are responsible for running around and doing all of the activities of the nest. So they gather the food, they take care of the queen, they rear the brood. They repair the nest or, clean the nest. But then there's soldiers. And so soldiers typically are involved in defense. But defense can come in many forms. And so there's sort of active defense. So things like army ant soldiers, right, with those big sickle-shaped mandibles that are used for defending the colony. But in turtle ants, they have something that's called passive defense. And so what they essentially do is they're responsible for making sure that no intruders or parasites get inside the nest. So how they do that is by leveraging their giant heads, which essentially look like they have dishes or saucers sitting on top of their head. And they just block the nest entrance of the little hollow twig they live in all day long.
Neat.
So all they do is act as a living door. And so that's their passive defense.
So, Corrie, if you were an ant, would you be a queen, a worker, or a soldier?
Oh, I would definitely want to be a worker. Come on. Everybody else has got a boring job. I mean, the queen, she mates once and never leaves the nest again. So all she does is become an egg-laying machine.
Right.
I'm like, the workers get to go out and run around in the environment.
Okay, so it's not bad to be a worker ant then.
No way.
That's great. Nels, do you want to move into our paper here?
Yeah, good idea.
I could spend an hour asking some questions.
I know. I was kind of waiting to see how many you could bring to get. No, it's great. So one of the things that made the timing of this recording kind of fun is that Corrie had a really interesting paper emerge just kind of the end of this summer in Nature Communications. The paper is entitled Comparative Genomics Reveals Convergent Rates of Evolution in Ant-Plant Mutualisms. And so we thought, in addition to just getting some of the great background and celebrating the beauty of these creatures, it would be fun to sort of put our money where our mouth is and actually traverse from some of the great ecology and the ant biology all the way through to the genomics that now is happening in Corrie's work, and then to try to understand how taking that genetic or genomic point of view might inform some of the behaviors and the biology going on here.
Great. I'm excited that we're discussing this paper. It was a lot of fun to do.
So the first author is Benjamin Rubin.
That's right. So Benjamin was a PhD student in my lab who's now graduated and is doing a postdoc at Princeton University.
Nice. Great place. Not too far from where I am right now.
That's right. Yes.
So when students do PhDs with you, you must be associated with an academic institution, right?
That's right. So I have an appointment down at the University of Chicago where I can take PhD students. It's in the Committee on Evolutionary Biology. It's a great program.
Nice. Wow, that's great. So what's the origin of this paper? What was the underlying background for it?
Yeah, so the underlying background is that there's been this long-standing sort of question in evolutionary biology about what drives genome evolution, right? Mm-hmm. And so from the standpoint of things that people that have worked on parasitic organisms for a long time, it's been well documented, and the sort of premise behind it has been well understood about why you might see increases in genome evolution when you have parasitic partners. And the thought behind that is sort of, the idea of a Red Queen's hypothesis, where, it's an homage to the Lewis Carroll book, Through the Looking Glass, that Lee Van Valen adapted into a framework on a hypothesis where he essentially used the quote from the Red Queen when she said to Alice, "It takes all the running you can do to keep in the same place." And essentially what they were saying is that when you have a parasitic system operating on a host's evolutionary framework, you might expect the two organisms to continue to escalate through time. So as the parasite tries to outwit the immune system or immune defense of the host's immune system tries to outwit the mechanisms by which it's being detected by the parasite, and so on. And so you sort of get this evolutionary arms race, if you will, where both organisms are constantly evolving at very quick rates to try to essentially one to outrun the other and the other to keep up. And so people have long thought about symbiosis as a continuum, right? So at one end you have parasitism, where one partner is being very negatively impacted on, through commensalism, where you might have both partners sort of having almost no effect to each other, to the other extreme, where you have mutualisms, where you have one partner or both partners are benefiting from this reciprocal engagement. And so, although we have a pretty good understanding about what the framework of molecular evolution looks like and the theory behind it for the parasitism, the other extreme of obligate mutualisms, no one's been able to explore it well, at least not in eukaryotic systems, because when it's been explored well, it's often been in microbial systems. And one of the things that we've seen, that as soon as microbes, in particular bacteria, move into a host and become an obligate mutualist, they immediately start chucking out whole portions of their genome, right? So, anything that they're no longer going to need to be free-living bacteria, they, essentially lose. And so that makes measuring, rates of genome evolution compared to free-living forms difficult. Mm-hmm. So we haven't been able to really tease apart what happens at a sort of, molecular or genomic level in these sort of obligate mutualistic systems. So Ben and I thought, well, what about if we could actually, take advantage of the fact that ants engage in many cases in obligate mutualisms with plants to ask this question. And so coming back to Nels's point where he said that, evolution often plays out the natural experiments for us, that's exactly what we did, is we leveraged that within this group of ants. Mm-hmm. So it's a single genus of ants. It's the genus Pseudomyrmex, and these are the ants that were made iconic from Dan Jansen's classic work on the acacia ants. And here we have ants that are engaged in obligate mutualisms with plants. And so the plant cannot persist through time without its mutualistic ant that acts as defense against any herbivores or encroaching vegetation. And in return, the plant provides nesting space and all the food resources that the ants need, either directly through things like extrafloral nectaries or food bodies, or indirectly by letting not that they let them, but the ants feed indirectly on the plants by farming other sap-sucking insects inside so that they'll get honeydew. And so
sorry, Corrie, can I just interrupt?
Sure.
I'm just super curious. So this idea of that mutualism, so if you take away these ants, how long does it take for the plants to either get eaten or outcompeted, or what's the tempo of that?
Right. So it depends on the size and the age of the plant. So young saplings won't last long at all. Fully mature plants actually may persist for a year, but not much more. And so they're usually, essentially, they succumb to, in some cases, competition, but usually herbivory is one of the biggest reasons for their demise.
I mean, obviously, the ants really get a lot out of this mutualism as well. But how dependent are the ants? So how long could they live without the the sustenance that the plants provide, either as a home for other microbes or directly in the sap?
In fact, so most of these ants are obligate with their plant hosts, so you actually don't find them in any other plants. Wow. So even though we could remove them and move them into another plant, we could probably actually, if we gave them an artificial diet, sustain them for a while. I don't know how productive they'd be as far as being able to produce sexual brood. I don't know that many people have done that experiment, but I do know that the ants are actually found nowhere else.
Mm-hmm. Mm-hmm. So I'm glad you brought up the red queen. So this is a hypothesis that, as you're pointing out, that a lot of people who think about kind of microbial evolution run into, especially in terms of the host parasite or host pathogen interactions. But I think it's also important to remember that, like, when Van Valen was proposing this idea, it actually wasn't microbial at all. It was much more ecological. And go ahead.
That's true. And actually, one of the things that I think really frames our work in thinking about the Red Queen is that he had multiple parts to his hypothesis. And most people actually remember that he said, you have these organisms that must constantly adapt to survive when pitted against ever-evolving opposing organisms. But I think the thing that people often forget is that he has this other line to it, in which he says, in an ever-changing environment. And so really it's that those two organisms are locked into this dance together, but they're also having to respond to all of the pressures that they're in their environment, whether they're biotic or abiotic, that are shaping the sort of this idea of a Red Queen hypothesis.
Mm-hmm, mm-hmm.
So you also mentioned this Red King idea, right? Can you talk a little bit about that?
Sure.
Did Van Valen come up with that as well?
No, so that sort of is a more recent hypothesis that's been put out there based on some theoretical modeling work, where because we didn't have systems where we could really go in and experimentally test what happens when you have mutualists engage in this kind of symbiosis, what happens at a molecular or genomic rate of change level. So people have gone in and done some modeling of what might in fact happen. And the idea behind a Red King is that you may expect to see one or more partners actually evolving at a slower rate while one's taking advantage of the other. And so that it's sort of this idea that you may see a slowdown in both partners, but more than likely what you'll see is only one partner is sort of at a slower rate of change based on the resources and interactions in the partnership.
So is it then still a mutualism? It seems to me that if one is taking advantage, it's not really anymore.
That's a great question. And so there's been a long-standing sort of, I wouldn't say argument, but conversation in the ecological literature about, where do parasites and mutualists evolve from? Because you could imagine that it's a slippery slope to transition between these things. And there's a whole body of literature out there that's been, looked at the rate of change from, how often do you switch between these states? Mm-hmm. And some people would argue that it's only beneficial to be mutualistic while it's beneficial to be mutualistic. And as soon as one or both partners can take advantage of the system, they absolutely will.
Mm-hmm. Mm-hmm. It's sad. It's the same thing with people. If you can game the system, people will do it. Same thing in the rest of the natural world.
That's right.
So sad. All right. So you set out to look at this in this paper, right?
That's right. And so one of the things we wanted to do was to let evolution play out the natural experiment for us, because being able to do these long-term sort of ecological experiments or molecular evolution experiments on eukaryotes is near impossible. Mm-hmm. So within the genus Pseudomyrmex, we have mutualism has evolved 3 times independently. So that really provides the framework where we can ask the question, if we examine the mutualist and its closest relative, which is a generalist, 3 times independently within a single evolutionary group, do we see signatures that are similar or not? Mm-hmm. And so, what would this tell us about the way that mutualistic behavior might impact genome evolution?
Mm-hmm. Right. And then you have something to compare to, which is really cool.
That's right.
And maybe do you have a sense of how these independent sort of acquisitions of mutualism, the tempo of that, like when did those diverge from the generalists? Is it roughly the same in each of those 3 examples?
It's roughly the same. It's on the order of magnitude somewhere between probably 4 to 8 million years. So in a pretty relatively short time frame.
Mm-hmm. And are there? I'm getting a little off track, but are there examples where that sort of switch from generalist to mutualism, like that that's happening sort of before your eyes, like that it wasn't 8 million years ago?
Yes, and so in fact, there's this beautiful study by Todd Palmer and his group at the University of Florida, where they've been working in a different species of ant plant mutualism in Africa, and these are the sort of iconic savanna acacias that are heavily, not preyed upon. I guess that might be the wrong word for the plant, but they're fed upon by the elephants and giraffes. And so it's long known that they have these ant partners that are a great defense against these large herbivores. So Todd and his group had this brilliant idea of, let's just build a fence around a bunch of trees and wait. And the fences only kept out the elephants and the giraffes so they could control for other things in the environment. And amazingly, what they saw is after 10 years, the plants stopped investing in the ant partners because they were no longer beneficial. They were actually a cost, right? So, and what I think is striking about this work is it's not just that the population changed through time. This is an individual tree. An individual tree stopped producing all of the nesting cavities for the ants to move into. It stopped producing so many extrafluorocnectaries or food bodies for the ants to feed on. So an individual tree without the selection pressure, and that comes back to that last caveat in Lee Van Valen's hypothesis about the red queen is that, the impact that the particular organism is, the pressure it's faced drives whether it's a beneficial or a parasitic interaction.
So do you think for any mutualism, if you remove the stress or the pressure, it would dissolve the mutualism?
I think in some cases, things like this, like the big herbivores, if we suddenly went extinct in Africa, we would have, I think the plants would kick all the ants out. Now, the ants would probably go extinct, which is unfortunate, but, the plants would thrive. I think in some cases that's not true. I think things like our gut microbes, I don't think that, I don't know what pressure would ever be taken away in a sense that one would ever persist without the other.
But you say the ants would go extinct, but those are the ones that have the mutualism with the plants, because there are other ants that don't do that, right?
That's right. So I meant the species that are only found with the plant.
The others would be fine. Okay.
So in this case, once the ants specialize or become mutualists, they sort of go down this one-way path where they can't sort of make a compensatory shift or adapt in a way to now go back to generalist. Is that the issue for the ants?
That's absolutely true. And so we see no cases of reversals within the ant plant mutualists, the group that we work on.
So these are interesting mutualisms because it's not one organism living inside of another, which you mentioned before, and there's a problem with losing genes, so you can't do this analysis. So are there other examples of, I don't know, ectopic, maybe we could call it mutualism, or maybe there's a real name for it, besides the ant plant?
Yeah, of course. I mean, there are lots of mutualisms out in the environment. And so, it depends on how obligate you want to define them. And so, of course, there are many groups of, butterflies that only can be found associating with ants, live inside ant nests, right? And what's interesting there is in some cases they've switched life history strategies, so they're no longer mutualist. In fact, the few carnivorous butterflies actually have evolved out of these mutualistic associations where now, once they've moved into the ant nest, they'll actually now start feeding on their hosts. Okay. And so, maybe that's another sort of example of that switch, that toggle between being a mutualist and being a parasite.
Right. Okay. So tell us how you studied this problem with ants.
Yeah, so essentially what we did is we went in and we sequenced a reference genome from within the genus so that it would be equally distantly related to our species pairs. And what's really great about ants is we got to leverage a little bit about their sort of life history strategy or their reproductive strategies in the sense that, you may or may not remember that ants and social insects, at least the Hymenoptera, have what's called a Haplodiploidy mating system. And so in this case, what happens is that once a queen has mated, she stores sperm in her body. And as she's laying an egg, she can unite the sperm and egg, and that will produce a female. So either a sterile worker, or eventually one can, in fact, become a virgin queen. Or if she passes an egg through her reproductive tract but does not release any sperm, it will develop into a male, and it's haploid. So it has half of the genome. And so that actually is something we leveraged in our genome sequencing, which makes things like genome assembly much more straightforward. And then, of course, we sequenced lots of other individuals for RNA to be able to annotate our genome. So once we had our reference genome, we then went in and to those three pairs of partners, where one's a mutualist and one's a generalist, we sequenced those genomes so we could map them back to our outgroup, our generalist outgroup species.
So for pseudo-Miramax, the genome had not been done previously?
That's right. And in fact, when we started this study, there were no ant genomes published yet. We knew there were a few in the queue. By the time that we were starting to analyze our data, there were, I think, 8 ant genomes had been published. But the closest one on an evolutionary scale is about 80 million years away. So we couldn't use that as a reference. I mean, it's still very distantly related. And so once we had those genomes, we could actually start to ask questions about, where do we see selection across the genome, and do we see different rates of molecular evolution in parts of the genome in these different species pairs?
And so since you had to actually kind of derive your own reference sequence here, maybe give us a sense of how much of an investment that was. How much time did it take, and how much of how many resources went into that?
Yeah. Well, in this day and age, actually, luckily, a lot of molecular biology is pretty accessible, and also even maybe financially accessible in a way that wasn't true even a decade ago. But this was the bulk of my former graduate student's dissertation work. He actually still has another part to publish on all of the microbes associated with this system, or at least the bacterial communities associated with this system. But really, he dedicated 6 years to sort of building up this project, and so it took a considerable amount of time.
Yeah, I just wanted to kind of highlight that because it's so easy for us to say. Like a lot of scientists are guilty of this, of, I'll just do the genome, or we just sequence the genome. and it's a sentence that kind of rolls off the tongue. But then when you sort of, peel back the curtain, there's so much of an investment that still goes into that. And I think especially, go ahead.
No, please.
Well, I was just going to say that especially as we continue to think about sort of exploring the genetic diversity of a lot of different organisms that we haven't considered yet, I think it's really easy to underestimate how hard that is and what an investment it is to do it well. And so
I couldn't agree more.
Yeah, and so the danger is that because we're so familiar now with genomes and have invested so much in the human genome, it really surprises a lot of people that if you just go to some of even our close relatives, some other primates, that the genomics is really in a state of almost disrepair because we sort of overestimate how easy it is to just apply what we already know from a very small selection of species. And so anyway, I just always like to point out that I
agree. And I think that it's a lot more challenging than people appreciate, that you actually have to have the material preserved in the proper ways to start with. So that's problem number one. But I also think that it does require a lot of resources, both in the form of the sort of chemistry and what it actually takes to get, usable and, high quality enough DNA to do the genomics. But on top of that, it does take quite a considerable amount of financial resources still to this day. And even though the costs are dropping, it still is not at the point at which, the average small laboratory can just say, well, I just want my genome sequenced. Now, the flip side of that, and I think one that is often really overlooked, is why do you need a genome? The number of times that people tell me like, I'm just going to sequence the genome of my, species of interest. And then I'm like, well, what are you going to do with it? And what's the question? Like, what are you going to learn from just having the one genome? And I think really, thinking about it in the framework of maybe something about the closest relatives and you're interested in targeting certain genes that you're of interest. But in many cases, I think that people are coming to this, like, it's easy to sequence a genome, so I'll just do it.
Yep.
Corrie, where did you collect the ants that were used for the genome sequence?
So in this case, some of them came from Florida. So I've had a long-term research program going there. And then others in this project actually came from Central and South America. So these species are interesting because the 3 independent evolutions of mutualism, even though they all happened within a single ant genus, interestingly, they haven't partnered with all the same plants. So one lineage actually is associated always with the sort of iconic acacias of Central America. But the 2 others are associated with 2 other plant genera. And so, in fact, across the 3 plant genera, they're from 2 different plant families. So it's not constrained on both sides by who they're partnering with, both the plants and the ants, but it is, within a single plant genus.
And the generalists were from the same area, is that right?
That's right.
So when you got one of the mutualists, then you would get a generalist from the same. I think you said.
In some cases, but the thing that was more important to us than the geographic origin of the species was having the closest phylogenetic or evolutionary related species so that we could control for time, right? Yep. And relatedness.
I think it's worth pointing out what you write here, that these generalists have very different behavior. They flee.
Oh my gosh.
They flee.
Yes.
Tell us.
Yeah, sorry. That's actually the major difference between them. So, anatomically, they look quite the same. They both nest within, plant twigs, but the generalists will be found in any sort of twig cavity they can take advantage of, where the mutualists are only found with their host plant.
Right.
And the major difference, as you've stated, is behavior. So the mutualists are incredibly aggressive. So they aggressively defend their host tree. They patrol it 24 hours a day. They have very different life history strategies when it comes to reproduction. So they'll actually defer reproduction, or at least they'll continue making more workers and not making new virgin queens or the sexual forms until their tree has grown large enough and they've been able to build up a large enough workforce to defend that tree, where the generalists actually have an incredibly different behavior and life history strategy. They actually are very timid. They rarely leave the nest except when they're foraging for food. If you disturb their nest, they flee. They actually won't even defend the nest. And their life history strategy, since their nests are so ephemeral, since they're not, defended in any way, their life history strategy is to go into reproductive mode within a few months of founding the colony.
Mm-hmm. Neat. Let me take a moment here to tell listeners about the sponsor of this episode, CuriosityStream. It's the world's first ad-free nonfiction streaming service. They have over 1,500 titles, 600 hours of content, and they were founded by John Hendricks, who started Discovery Channel. So you're guaranteed access to real science shows. You can watch these films on your web browser or on any of those appliances that connect the internet to your TV, like Apple TV or Roku. You can get it in 196 countries. What they have is a wide variety of science, technology, nature, history, documentaries, interviews, lectures, all good stuff. For example, Stephen Hawking's favorite places. He pilots a fantastical CGI spaceship across the universe, stopping off at the places that he thinks are really cool. How neat is that? Another one called Digits. This is a 3-part series hosted by Derek Mueller, who's creator of the Veritasium Science channel on YouTube. And the series features interviews that have never been aired before with Ed Snowden and Vint Cerf. Everyone knows Edward Snowden. And Vint Cerf, of course, co-founder of the internet. Not what's his name? Who was the vice president who said he founded the internet?
Oh, Al Gore.
Al Gore.
Yep.
Yes, it was not Al Gore. Deep Time History, a 3-part series on the 14 billion year history of the universe. Underwater Wonders of the National Parks. A lot of cool stuff. They also have a pretty big 50-hour ultra-high definition library, 4K, which you can see as well, and that's growing as well. Now they have monthly and annual plans, and they give you a lot of savings if you take a year out at a time. They start at $2.99 a month, less than a cup of coffee, or the cost of a single title on competing platforms. Check out curiositystream.com/microbe. Use the promo code microbe when you sign up to get unlimited access to the world's top documentaries and nonfiction series, completely free for the first 60 days. That's 2 entire months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign up. We thank CuriosityStream for their support of TWiEVO.
Yeah, that's right. Lots to be curious about, and feed your curiosity even more. So Corrie, I'm curious. You have you now have your genomes.
That's right.
And so remind us the question that you really wanted to go after, the differences of rates of evolution.
That's right. So we wanted to ask the question, what happens when you have species that engage in these obligate mutualisms? What happens to their rates of genome evolution? Do we see that they have a similar pattern as seen in these parasitic or antagonistic obligate mutualisms where we have increased rates of molecular evolution? Or do we potentially have a slowdown as in one or of the partners as predicted by some parts of the red king hypothesis, pardon me. And so we really wanted to be able to tackle that. So now that we had full genome sequences, we could actually ask this question. Now,
yeah, and so when you say rates of evolution, what are the measures? What are the things that you want to compare?
Right. So we want to see how many sort of changes in nucleotides or in the letters of the DNA do we see in those pairs compared to the outgroup species, so the more distant relative. So we're making those those pairwise comparisons between the mutualist to the outgroup and then the generalist to the outgroup. And since those are each other's closest relatives, we know that they're the same age on an evolutionary timescale. So we're controlling for time because we know that there's some constant rate of mutation that's always sort of happening. So we're controlling for that, and then we're saying, okay, how much additional change do we see across the genomes, compared to one another?
Mm-hmm. And one of the other things that you bring up in the paper that you have to sort of be concerned with when you're thinking about rates of evolution like this is the demography. So remind us how you account for this or consider this.
Right. So one of the major findings of the paper is that we do in fact see increased rates of molecular evolution across the entire genome. Now, as you highlight, there are sort of demographic reasons that we might see these rates of change. And so some of them are things like generation time. And so maybe the mutualists just have consistently larger shorter generations times, so they would just have more rates of change. So, the way to think about that maybe is that if we compare the amount of rate of change of a fruit fly compared to a human, some of that's just driven by how many generations can co-occur in the same time frame. Mm-hmm. So if that's in fact the case, what we would expect to see is that if what's driving the faster rate of molecular evolution in the mutualists, it would just be that they had shorter generation times. They had more time in each successive generation compared to the generalist. And that's not what we see. And in fact, I already sort of talked about the fact that that has to do with their life history strategies. Mm-hmm. So the generalists go into reproduction within about 2 months of founding a new colony, where the mutualists typically, defer that for about a year and a half. So a significantly longer period of time before they actually engage in reproduction. And that's one measure, but of course there are additional ones. And the next one, of course, could be something like population size. Mm-hmm. And so in this case, maybe it's just that the mutualists have, smaller population sizes, which would drive a higher rate of molecular evolution when we make these pairwise comparisons. Mm-hmm. And so we did a series of tests to address this question. And what we see is that we have, no signature that suggests that the mutualists have smaller population sizes. And in fact, in the few cases that we looked, it seems to be in the opposite. So we can rule out population size as potentially being one of the mechanisms to in this pattern that we're seeing. There are some additional things that we can look at, one of which is relaxed selection. And there, again, what we might see is a frequency and repetitiveness across the genome in our mutualists. But we don't actually find any consistent differences that suggest that that's what's driving the rate of change. And so although we can't officially rule out relaxed selection, it doesn't seem to be what's driving the patterns we see. And the last one that we explored was mutation rate. And so in this case, maybe what we have is just a higher frequency of mutation occurring in the mutualisms. And when we did look for this signature across the genome, we do find a trend towards higher rates of mutualists. So again, maybe elevated mutation rate might explain some of what we're seeing, but it doesn't seem to explain all of it. So it really comes back to is potentially behavior driving this, or something about this relationship, this mutualism that's driving this, pattern that we're finding.
Mm-hmm. So you think that perhaps DNA repair proteins are involved in this, right? Can you tell us about that?
Yeah, so we actually also did some tests for sort of positive selection across parts of the genome to sort of say, what might be explaining the pattern, both in the increased rates of molecular evolution, but also, when we started out this project, we had some hypotheses that we thought, where might we see positive selection across the genome? And interestingly, we didn't find signatures in some of the places we expected. And so, we were talking before about one of the things that's so different about them is their behavior. And in other Hymenoptera, researchers have been able to show that there are these aggression-related genes. So we thought, well, great, this is, maybe exactly what we'd expect to see, that we're going to see strong positive selection on these aggression-related genes in our mutualists since they're so much more aggressive. And we didn't find that. So, there are parts to the story that, didn't go as, predicted, which is exactly the way science always works. That's right. Which is part of the fun of it. But, one of the things that we did look for is, when we saw that we had these overall increased rates of molecular evolution, could things like DNA repair changes be affected by the mutation rate? And interestingly, we do see that some parts of the DNA repair mechanism have convergent changes in parts that may explain some of the fact that we see this faster rate of mutation.
So
now, we haven't been able to actually experimentally test that, right? So it'd be really cool to be able to go in and do that.
Yeah.
One of the things I've learned on TWiEVO is that it's not always the gene that mediates change, right? Right, Nels? So it can be control sequences, right? Did you look at that? And so maybe these aggression genes are all the same, but their expression levels are different?
That's right. So we didn't do that. We didn't have enough sort of samples to be able to go in and look at that in any, as far as like we want to look at gene expression then, and we'd want to probably do some controls in the environment, right? And so we haven't done that yet, but that I think one of the things that I'm excited about is that this study provides a framework for other mutualistic systems to say, do you see similar patterns? But also within this system, there's a ton of questions left unanswered. I mean, one of the things is that if many of the changes we're seeing are related to behavior, you might expect to see significant expression differences in genes that are related to the brain. So, if we went in and did all kinds of, brain dissections, could we sort of tease apart, what's driving the difference in behavior between the mutualists and the generalists?
Yeah, no, it really is an interesting question, and one that's could be pretty, of course, it's hard to get a handle on. So, for example, looking at the aggression genes for signatures of positive selection. So certainly, if we consider from a host parasite or a host pathogen perspective, a lot of the genes that we see under strong positive selection, these are cases where there's some, protein-protein or molecular interaction where you have these, temporary advantages on either side. And so as a consequence, you end up getting these kind of a phylogenetic signature where there are fixed changes in protein sequences. And so basically as an outcome of the fact that this is so repetitive, that's where the kind of DNDS value becomes really useful. It seems like the challenge with the aggression hypothesis there is that you could imagine a scenario where, like Vincent's suggesting, if you just change the expression, or maybe it's one it's one mutation that can, on one occasion, sort of flip the switch for the ant going from more, I guess, docile to becoming super aggressive. That, that's the challenge in this case, right? Is to match sort of the measure with the behavior or the phenotype in that case. And so
that's absolutely true. Yeah. And I think coming back to that, I mean, part of it was we used a very conservative approach in this paper because we were interested in sort of the sort of shared mechanisms. So we only considered situations in which we saw strong positive selection in all three mutualists. Yeah. So it could be independent mechanisms that are driving the behavior or, some aspect of the biology that we would miss in our conservative approach. So you could certainly go back in and, farm these genomes and ask the question, if we just make the pairwise comparisons, do we see, I'm sure we see a lot more genes under positive selection. Now, actually knowing their function in the host is not always as straightforward.
So in getting back, I totally agree, Corrie, this idea of the power of the independent course of that evolution and using that as a way to triangulate onto sort of shared changes. And so it sounds like you uncovered a few of those. So wasn't there one, for example, for myosin heavy chain? Was it where you saw something?
Yes, which was really, interesting. It wasn't one that in fact, when we found it at first, it was like we were puzzled. Like we kept trying to get our head around it. And we tested it a few different times to make sure that, it wasn't a false positive in any way. And we still don't exactly understand why. But, what we sort of hypothesize is that, these mutualist species, they're active 24 hours a day. Their muscles are constantly in activity, unlike what we see in the generalists. But, seeing these, I think our strongest signatures of positive selection that were shared among all the mutualists had to do with myosin heavy chain.
And you have no idea why, right?
Well, we don't know for sure, but, again, it would be one of those things that, being able to go in and do some manipulative experiments, like maybe knocking them down or knocking them out, maybe seeing, or do we see expression level differences based on activity or timing of, development, because we know that ants have different roles within the nest as they mature. So you may actually have some of these things switched on later in development, like, we see the age polyethism like we see in bees, right? So that an individual has a different role within the nest based on the age of the individual. And so, yeah.
Maybe the generalists, they run away when they're threatened. Maybe that's Miles.
Yeah, it's a sort of a short burst, fast twitch reaction.
Right.
Yeah, exactly. That's absolutely true.
No, but it is cool that you just, these surprises that come up then sort of form the basis of a new hypothesis that with increasingly tractable genetic tools to go in and manipulate possibly like that gene and then maybe sort of reintroduce it back into the behavior of the mutualists. You have like that sort of a glimpse at the future for the first time to be able to do that.
That's right. We're getting there.
Can you genetically modify ants?
Well, it depends on what you mean by genetically modify, I guess. So if my answer, of course, is yes, but, it's sort of what level, it's not always is more straightforward. So, I think one of the reasons that ants haven't sort of become this laboratory model system, despite the fact that they have social complexity and these really interesting social behaviors, is the fact that, one, getting them to breed in the laboratory is quite difficult, and two, their generation time. So even though we see individual workers, being sort of born every day, you have to keep in mind that the true generation time is the time from a queen to her first offspring that are sexual forms. And in some species, that can be up to 5 years. And so generation time of 5 years for a laboratory system is not very tractable. But there are species that are now being kept in the laboratory of ants. I have to admit that my colleagues are going to kill me if they listen to this podcast. But most of the species that have the fastest generation time and are amenable to living in the lab have the least interesting biology.
We'll put out a special edited version for anyone who knows about ant-ecology.
That's right. Listen to this one.
Yeah, right. But are those examples like the leaf cutting ants? Aren't there experimental colonies of those? I guess I'm thinking of like maybe Cameron Curry up in Madison and sort of in your neck of the woods.
That's right. And so they're really interested in microbes within the system. And so they can actually have a single colony, right, that, can live anywhere up to 30 years. And so really in that system, they're not manipulating the host as much as they're interested in the microbes within.
Gotcha.
And the fungi that they're farming, right, which is another microbe in the system. And so I think they don't necessarily need to have the host in a tractable system of, genetically tractable systems. So they just need the host to sort of persist to maintain the things that they're interested in.
Gotcha. And so in that case, the manipulations are on the microbes associated with the ants, and that's where you can sort of get some more experimental traction. Yeah.
Absolutely right. Yeah. But, one of the things that, despite the fact that they have these long generation times, we have done some experimental work in my lab where we've looked at things like, can we shift bacterial communities within the guts of ants based on the diets that we're giving to them, which also gets at the question of where are they acquiring some of these gut microbes? Is it something they just reacquire from the environment all the time, or are there actual stable core community members of the guts? And so even though we can't sort of do the manipulations at the host level all the time, we can still get at questions that inform us about the biology of the host.
How complex is the gut microbiome of an ant?
It depends on the species of ant. One of the things that we're finding is that things that live at the extremes, so things that are herbivores or vegetarians or things that are obligate predators, seem to have core bacterial communities that are constant through time. In the case of the herbivores, in fact, many of them are co-evolving with their hosts, and some have simple bacterial communities with just a couple members. Some actually have very complex bacterial communities in their guts with, 15 to 20 members that seem to be persistent through evolutionary time at the scales of 50 million years. Now, interestingly, the species in the middle end of that spectrum, sort of all of the generalists, many of them seem to have almost no bacterial communities associated with their guts, which, flies in the face of us thinking that everything has a gut microbiome. But maybe if you have a diverse enough diet, you're not going to need to have bacterial partners that help, upregulate your diet in some way.
So they have zero bacteria in their gut?
I mean, they don't have zero, but they essentially have nothing that we see as constrained through time. So the density in proportion is low, and that if you sample individuals through time and space, that it doesn't seem to be that there's a member that's always there, right?
So if you deplete the gut microbiome of different ants, does it have a variety of effects?
Yeah, so we've done a lot of those experiments, partially because we want to be able to ask questions about the effects on host fitness, but in some cases it's also that we want to look at the function of the bacteria itself. And maybe not surprisingly, when we give lots of antibiotics to ants, they don't do very well for that long. But interestingly, we actually part of a collaboration I have with several of my colleagues, we've been able to culture some of the core microbes of the turtle ants for some, questions about the functional role of those bacteria so we can do assays of them, but also so that we can get pure culture lines so that we can sequence individual genomes. We've been able to do some metagenomics, but we'd like to be able to actually look at individual ones. But for me, what gets exciting is now if we have this capacity to knock out their bacterial communities with antibiotics and culture the members directly from their guts, the strains and different bacterial partners, can we reintroduce them individually and or in combinations to rescue host fitness? And so that's what I'm getting excited about. That's still probably a year or maybe more down the line, but we're getting there.
Nice.
Yeah, that's cool. So getting back to the paper here, so we already kind of hinted at the sort of some of the shared surprises that emerged from the genomic analysis, the increased rates, the heavy chain convergence. Were there other patterns that you saw?
Yeah, interestingly, one of the things that we saw, and maybe not surprising, was that we actually see signatures of positive selection on genes related to the nervous system. So genes engaged in neurogenesis and neurotransmission. And so if really the thing that makes these ants fundamentally different is their behavior, these would exactly be the kinds of genes you'd expect to see positive selection on. And so maybe it wasn't a surprise. It was actually kind of almost like reassuring in a sense. But for us, the big surprise was the rates of molecular evolution. So we'd actually predicted we'd see a slowdown in the rates of molecular evolution in these three lineages that are mutualists, obligate mutualists. And that's exactly the opposite of what we saw. And interestingly, it doesn't just pertain to the protein coding parts of the genome, it's to the entire genome. We see overall faster rates of molecular evolution.
So, and that's as measured by things like the transposable element content or gene duplications? What are the other?
So we have looked at things like transposons, but in this case, really it was sort of the mean sort of rates of molecular change, those nucleotide changes in those pairs, those three different pairs to the outgroup sequence, right?
I see.
So we also looked at rates of DS and DN and again see the same pattern. In DS, we still see elevated rates in all of the mutualists compared to their closest generalist relative. And in two of the three cases, we see that pattern within the DN ratio, DN in this system.
The rate. So you're talking now about the rates of non-synonymous codon substitution. So this sort of property that I guess emerges from the degeneracy of the genetic code, the fact that you have these codons, and then if you mutate one nucleotide in the triplet that codes for amino acid, that then you have two outcomes. One being that mutation is a non-synonymous change that actually changes the amino acid, or silent or synonymous, the DS, the rate of the silent ones,
right,
where the DNA mutation, it still encodes for the same protein. So in that case, how many, I think you say, was it something you were comparing across something like 4,000 genes? Is that correct, in these pairwise comparisons among the three? Yep.
That's right. So we looked at a little over 4,000 genes to look at, do we see, one, just at the level of the entire coding region, do we see overall faster rates? But then we could actually go in and ask questions about those synonymous and non-synonymous changes as well. And again, the pattern holds that the mutualists have overall faster rates.
Yeah. And interesting mystery.
Yeah, it's a mystery. Now, I mean, one, I guess, naive idea would be that there's less selection pressure on the mutualists, right? They don't have to deal with as many issues.
Well, maybe, but I don't know that you would necessarily expect genomes to increase in rates if that were the case. I mean, I don't actually know that there is a prediction, but the fact that we see the same pattern in all three lineages, I think, is the striking part of this story. I think if we only had the one pair and we found this, it would sort of be like a just so story. Like, okay, well, we see an increased rate. We don't really know what it means. We can't rule out that it's not some factor of the environment, or some aspect of the unique biology of this organism. But the fact that we see the same pattern across three different evolutions of a behavior is, I think, really the compelling part of this story.
Mm-hmm. Yeah. And actually, do you think there might be sort of an echo in that pattern on the other side of the mutualism if you looked at the plants? Or are those species too different, or you just can't make those comparisons? I'm wondering if that's like a two-way street, basically.
Right. So my prediction would be that species that engage in obligate mutualisms, we expect to see similar patterns that we're finding here. And maybe it's that fact that toggling between being an obligate mutualist and an obligate parasite isn't actually all that different. Now we don't know the answer from the plant side. I would predict we'd see faster rates of molecular evolution in the plants as well, compared to their closest relatives. Now, since these plants aren't each other's closest relatives, we can't do that sort of nice evolutionary experiment like we have here. Mm-hmm. But one of the things that I think is interesting is that there's really only ever been one other study of 2 eukaryotic partners that's found something similar. And that actually is a paper that came out in, I think, the early 90s that was looking at a very short piece of DNA. I think it was like 1,500 base pairs of a ribosomal gene. So not something that was necessarily coding for something in particular. And in sequencing that short piece of DNA in lichenized and free-living fungi, what they showed is that all of the fungi that engaged in the symbiosis, right, in this case with algal partners, had faster rates of molecular evolution in that very short piece of DNA. And in that paper, what they said was, well, we don't understand why this is. They actually did not attribute it to the behavior of the mutualism at all. What they attributed it to was that the lichenized fungi grow on rock surfaces and are exposed to things like solar radiation and forces from the abiotic environment, and that the free-living fungi were living deep in the soil and were, not exposed to these same pressures. But maybe it's just if we go in and sequence a bunch of genomes of these lichenized and free-living form fungi, maybe that'll be another system to really test. Do we see this faster rate of genome evolution across the entire genome, or is it something that was just unique to that short fragment that they sequenced?
Yeah, interesting.
And I'm thinking these changes in the DNA repair proteins, that may be the key to it. Those were, selected for because they give you an overall increased rate. And that's somehow it's beneficial for the mutualists, right? And Nels, I was thinking about viruses, which are obligate parasites. And they have a wide range of mutation rates, though, depending on the genome.
Well, that's right. But I think that can also reflect, I think we need to be careful thinking about what we define as a mutation rate. So if you're saying, I think generally we limit that to the point mutation rate, where you're, of course, you're 100% right. But then, as we think about all of the other mechanisms of diversification or mutation, recombination, a topic that we've hit on in TWiEVO a few times now, and even other mechanisms that, it would be interesting, I guess, whether that sort of rebalances the scales a little bit. If some, if a slow evolving virus as measured by point mutation rates is actually sort of a screamer for high recombination rates.
That's true.
Yeah. Well, we do know for many viruses, if you improve fidelity, it's not good.
Yeah, exactly. That there is a real tuning there.
Yep. Even bacteria as well, for sure. Mm-hmm. Because everybody has an error-prone polymerase. If you fix it, it's not good. Mm-hmm. Cool. Anything else from you, Nels?
No, this is really fun. I like how you ended the paper too, Corrie. I think on a striking sort of a good note here about how you're sort of glimpsing at these patterns and using the power of the three comparisons, but then I think also accounting for the fact that there is so much going on necessarily, right, in these systems, that to positively disentangle all potential mechanisms underlying the observed differences in the rates of substitution is hard to do, but that this sort of provides sort of a first foothold to be on the lookout for these kind of patterns as people start to consider and study other mutualisms. It just feels like we're kind of at the exciting start of an era where we can start to think about these patterns, observe them for the first time, and then start to form hypotheses and dissect what this might really mean for evolution of these more complex behaviors.
I agree, and I think taking a comparative biology approach is going to inform us a lot about, different aspects of biology. And it comes back to that idea about, thinking of museums as living, facilities that tell us about life on Earth, both contemporary life on Earth, but even deep through time, and really sort of thinking about all of the questions we ask in biology and in the life sciences in the context of a comparative biology approach, like what have we learned from nature? What can we learn from nature? I think is where we're going to see some really interesting research come out in the next few years, and even related to things like human medicine and human health and crop science and all of those things. I think we will begin to realize that asking questions in isolation isn't going to give us the most important answers.
Mm-hmm. Or it's just one set of answers, but that there's more to be learned from the comparative approach. And I mean, for me, that's what really takes a museum like the Field Museum, other natural history museums, and makes it more than just sort of a relaxing afternoon, but becomes like an inspiring place to think about new ideas and to move forward in exciting ways.
I couldn't agree more.
All right. Corrie, the last thing we do on TWiEVO is what we call a science pick of the week. Nels and I pick something to share, and we'll do ours in case you want to think about something. You're welcome to find something.
Yeah, this is our like pop quiz part of the podcast. So you have about a few minutes. It'll probably take us about 5 minutes of meandering through our comparing notes on our picks. But if you think of anything that you want us to highlight, just let us know when we get to that. Yep. So, Vincent, this week, my science pick of the week is trying to get a little bit at what I think are interesting collisions between art and science. So how do you take a concept like evolution that's sort of this massive theme of all of biology and then capture that in sort of an artistic way? And so I found this animated GIF a while ago, and I actually use this slide in some of my lectures to grad students and undergrads as just sort of a way of breaking the ice as the first slide as people are kind of coming into the room. Mm-hmm. And so, and then to have people kind of look at it, consider it, and even just try to explain what they might be looking at. And so this is a quote unquote mind-melting animated GIF of the process of evolution by Nicholas Fong. And it's highlighted at a website here called boom.com, and there's about, it's 7 O's in boom. We'll put the link up on the website. But so if you click over there, and I think you can see this first one, it's this sort of spiral of colorful red and green patterns. And so what this is meant to do is to sort of capture the evolutionary progression from single little microbial organisms out to humans as the GIF sort of progresses in this sort of spiraling pattern. Do you have it up on your screen?
I do. It's amazing. I love it.
It's kind of mesmerizing.
Yeah, you start at the middle and you follow things up and watching them differentiate. It's pretty cool.
Yeah. So it's both artistic. Cool. Yeah, but it also kind of captures, I think, some information in a really interesting way. And then you get to the very edge of the spiral as it progresses, and then there's sort of something that looks like a human or hominoid that spits out a microbe and then sort of dissolves. I don't know if that's if that's meant to be a commentary on our sort of impending demise as a species, or what. But so anyway, and then if you scroll down, you'll see other less biological animated GIFs by this artist, but kind of on the same style or theme. And there's some just really kind of fun to the way he kind of captures these things and the motion to them and the patterns in them. So anyway, that's my pick of the week. It's sort of a collision between art and science.
Very nice. Plus, I'm exploring some of these other links at Boom. They have incredible photos. Look at this. If you just go to photo, vibrant overhead portraits of Vietnamese street vendors. They're gorgeous. Wow. They're all the same thing, an overhead portrait of people bicycling big tubs of stuff, vegetables, and so. Oh man, they're just beautiful. What a sight! Could spend some time. Could waste some time here, Nels. I think
so. Yeah, it's sort of a rival to Curiosity Stream.
Wow.
Is this boom.com? Yeah. So anyway, that's my pick. How about you, Vincent? What are you?
Well, my pick is a website that I built for my colleagues here. It's called Parasites Without Borders. It's all one word.com. And it's put together, so I was the webmaster, but Dixon Despommier and Daniel Griffin, who I do TWIP with this week in parasitism, put together this site. And the idea is to engage the world about parasites, which don't have borders of all sorts, and it's an informational site. They want to disseminate information. They're eventually going to have lectures here, and they've just revised their book, Parasitic Diseases, 6th edition, and you can actually download a free PDF of it on this site. You can also buy a hard copy if you'd like to support them, but you can download it. And it's all revised from the last one, which Dixon and others have done. So it's good just for that if you're interested in various parasites. Very comprehensive book.
And it looks like under the link, the problem, some of the main parasitic players with big implications for our species: malaria, trypanosomes, leishmania, and on and on. Yep.
Right. So they are. This is an educational site, and it'll grow, but they want to make more awareness. That they want every medical student in the country to have a copy of this book. They want to, put this information into medical schools because they feel that medical schools don't teach enough parasitology, and it's really important. I think it's a good goal, and I love the site, and I like the information, and particularly you can get the book there, which is really great, and we deal with that on TWiP all the time. So check that out.
That's fantastic.
That is.
We have a listener pick from Chris. He writes, "Hi, Vincent and Nels. For those who would like to see the shark weapons, check out this episode of The Brain Scoop from Emily Grassley at the Field Museum." So I thought this is appropriate because...
That's absolutely perfect, because the Brain Scoop was going to be my science pick of the week.
Nice. Now, Emily's not at the Field Museum, is she?
She absolutely is. We scooped her up and hired her to come and do all of our science communication through her Brain Scoop YouTube channel on the research that actually happens at natural history museums.
Oh, that's cool. Wow. Now, Nels, what's the shark weapons? Does that have something to do with Josh's opinion?
Yeah, that's exactly right. So if you remember, Josh was talking about these ideas of historical ecology. So going back into the museum collections and sort of looking at them from a different perspective too. And so he was talking, and I forget the exact location, but these swords, I believe, that had shark teeth as part of the decorations on them, on those. And that the ecological question they were able to address was the fact that they knew when those swords were fashioned, and then could figure out the species of shark that the teeth were taken from.
Got it.
And that actually allowed them to realize that there are actually species of shark swimming in those waters at a time when it wasn't thought that they were there. So that was that was sort of the discovery. And so this is cool because now we can actually connect that together with what these things look like themselves.
Neat.
Very neat.
All right.
And I have to say, I cannot give enough props to Emily Grassley and her ability to take complex scientific questions and research that's happening behind the scenes at museums and portray it in both a really entertaining but also informative way. And so if you guys have not watched The Brain Scoop, I just want to tell you now, you're going to have to take an entire couple of days off to go and watch a bunch of the episodes. They're all really great, but there's a few that really stand out in their ability to sort of convey, work that's happening at museums, either from a historical standpoint or even from the context of, what are we learning from collections? And so two of my favorites actually are, one is called The Flapper and the Panda. And it's really interesting from both the standpoints of thinking about how to, historical collections come to be in museums, but also the role of women in early science. I think it's a good episode. And there's her most recent one is called How Bird Vomit Helps Us Understand History. And it's a really interesting episode, thinking about how we can use historic collections to understand the impact of humans across entire landscapes. And so
nice.
I'll let you guys go and watch those.
That's perfect. That's exactly what a science pick should be. Thank you. Thanks, Corrie.
You got it. Yeah, it's been my pleasure to be here.
So this episode is going to be on iTunes, microbe.tv/TWiEVO. And if you have questions or comments, send them to TWiEVO at microbe.tv. Consider subscribing either in iTunes or on your favorite iPhone or Android device. It's free all the time. And if you want to support our work here at TWiEVO, you can go to microbe.tv/contribute. We have a number of ways you can help us. Patreon. You can use your credit card or PayPal to give us some money, or you can make purchases at Amazon. We have an associates program, and a little percentage of your sales comes to us. We also have a store where you can buy TWiEVO swag, mugs, and T-shirts, and mouse pads, and all that stuff, and we get a little bit of those proceeds. So check out microbe.tv/contribute. Nels Elde, you can find him at cellvolution.org. He's also on Twitter as L Earlybird. Thank you, Nels.
Thank you, Vincent, and thank you, Corrie. This has been really fun. And I was just thinking, if you guys are talking offline about starting one of these this week in insects, I already have a catchphrase for you.
What's that?
Twins this week in insects.
Twins. That's it. This week in insects.
All right.
I love that idea.
I'll leave it there.
Good, great. Corrie Moreau is at the Field Museum. You can also find her on Twitter, Corrie Moreau. Thank you so much.
It's been my pleasure. It's been really fun. Thanks so much.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsor of this show, Curiosity Stream. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious. Nice office. Cool. You got a lot of cool stuff. A big ant. Well, now it's too bad you couldn't go visit.
I know. I'm still kicking myself for that. I was just there last week. Next time.
If you go, let me know. I'll come with you.
Yeah, exactly. Field trip.
It'd be fun to show you around the museum.
Yeah.
Awesome.
Yeah, I don't go to too many museums. Especially,
what?
I just don't.
You live in a place with amazing museums.
You know what they say, wherever you live, you don't do stuff there. Yeah.
I know what you mean, actually. I have to force myself to go and do cultural things around Chicago at times.
But science is changing, as you know, museums, because it used to be there were just collections, right? And now people do molecular biology in museums. So it's getting...
I know. It's awesome.
It's getting very compelling. I mean, here, the Museum of Natural History, we have several faculty members there who have joint appointments here, and they're sequencing things, and it's like, whoa. It's no longer dusty collections, right?
That's right. It's really true, actually. And I think this whole movement in molecular biology, and especially now that we have these technologies that allow us to take advantage of even in many ways degraded DNA, it's sort of unlocked this treasure trove of questions we can ask with historic collections. That's
great. Taking advantage of degraded DNA. Well, we had a guy from Columbia, Josh Drew. I don't know if you know that name.
Yep, he actually did a postdoc here first.
Yeah, that's right. That's right. He knew you or knew of you. He takes advantage of museum collections too.
That's absolutely
it's just great.
right.
And I think, Nels, we're going to encounter that more and more.
I know, I love it. I also think these museums, at least for me, as a whatever molecular biologist, whatever I am, it just feels like an oasis for science. Like you just go into these beautiful spaces and just sort of draw your breath and relax and just enjoy it's just a celebration of all this stuff around us. So it's just for me, it's really rejuvenating to spend some time at a place like the Field Museum, other museums of natural history. We've got a good new one here in Utah.
That's right. You guys do.
Yeah. But hey, listen, this conversation's already running pretty hot. I think we need to start taping something.